just rushes out to dilute the external environment.
And the cell instantly dehydrates, shrivels up, and dies.
So how do halophiles survive that osmotic pressure?
They counter it through the accumulation of what we call compatible solutes.
Compatible solutes.
Yeah. So instead of trying to pump the salt out, which would be impossible given how concentrated the outside is, they artificially raise their own internal pressure.
Oh, that's clever.
They synthesize and stockpile massive amounts of specific organic compounds, usually unique sugars or amino acid derivatives, directly within their cytoplasm.
So they basically pack their own interior with these solutes until their internal osmotic pressure perfectly matches the crushing osmotic pressure of the salt water outside.
Exactly. The pressures reach a state of equilibrium.
Wow.
So the internal water has no thermodynamic incentive to leave the cell. This allows the halophile to retain its hydration and function totally normally in a hypersaline bath that would turn standard organic tissue to absolute dust.
This just proves that life possesses this incredibly sophisticated toolkit for maintaining internal chemical order against massive external chemical chaos.
It really does.
I mean, if biology can engineer a way to pump protons against battery acid or balance osmotic pressure in a lethal brine, then we just cannot dismiss the highly acidic, toxic clouds of Venus.
Or the concentrated subsurface brine networks we strongly suspect exist on Mars.
Right. We already know the biochemical workarounds exist.
We've seen them in action.
But so far, we have been looking at highly specialized, single-celled organisms, right?
Yeah.
Like the hyperthermophile is a master of heat, but it would die in the ice. The psychrophile loves the cold, but would absolutely boil at the thermal vents.
They are ultimate biological specialists.
But the natural question that comes up is, does a biological generalist exist?
A great question.
Is there an organism equipped with basically a biological Swiss Army knife, capable of handling all of these extremes simultaneously?
And this leads us perfectly to the tardigrade.
The water bear.
The famous water bear. And I really want to highlight a massive distinction right away here.
Okay, what's that?
The extremophiles we've discussed so far are archaea and bacteria. They are single-celled, but the tardigrade is a complex, multicellular animal.
Right, it has 8 legs.
It has 8 legs, it has muscle fibers, it has a digestive tract, and it even has a central nervous system.
And multicellularity usually introduces a lot of fragility, doesn't it? Like complex systems just have more points of failure.
They do. Yet the tardigrade possesses survival statistics that frankly defy all logical biological parameters.
It's alien.
They survive via a mechanism called anhydrobiosis, which literally translates to life without water.
Okay, so when a tardigrade is exposed to a severe environmental drought, what does it do? Because it doesn't attempt to fight the dehydration, right?
No, and it doesn't flee either. It initiates this highly orchestrated total system shutdown.
A shutdown.
It forcibly expels up to 99% of the water from its body. It pulls its 8 legs inward and compacts its entire body into this really dense, shriveled, microscopic ball known as a tum.
TUN, right?
Exactly. And the tun state is a profound metabolic standstill. I mean, the organism's metabolism drops to less than 0.01% of its normal rate.
That is practically undetectable.
It really is. But the true marvel is what is happening at the intracellular level here.
Because water provides the critical structural scaffolding for life, right? Like it holds proteins in their folded shapes and it keeps the DNA double helix stable.
Exactly. If a multicellular animal loses 99% of its water, the proteins crash into each other and denature, the membranes collapse, and the DNA fragments. It should be lethal.
So to prevent this total structural collapse, what does the tardigrade do?
It replaces the expelled water with a unique, highly specialized sugar called trehalose, along with some specific intrinsically disordered proteins. These molecules flood the cells and essentially turn into a biological glass.
A glass.
Yeah, it's a process called vitrification. The sugar matrix basically coats every critical molecule, the DNA, the lipid membranes, the protein structures, and just locks them securely in place.
It acts like a molecular concrete.
Exactly. The organism's interior transitions from a fluid state to an amorphous solid.
Okay, imagine you have a delicate water balloon. If you puncture it and drain the water, the rubber just sags, folds in on itself, and crumples, right?
Right.
But if you slowly drain the water while simultaneously injecting a liquid glass that hardens instantly, the balloon retains its exact perfect shape.
That's a perfect analogy. The internal structures are physically frozen in place, preventing any physical or chemical degradation.
And once locked in this glass-like ton state, the tardigrade becomes virtually indestructible.
The parameters of its resilience in this state are almost absurd.
Give us the stats.
Well, they can withstand temperatures plunging to minus 272 degrees Celsius.
Which is just a fraction of a degree above absolute zero.
Right. And on the other end, they can be subjected to temperatures exceeding 150 degrees Celsius.
So well above boiling.
Way above. They survive hydrostatic pressures 6 times greater than the deepest ocean trenches on Earth. And they can remain in this state of suspended animation without aging at all for decades.
They just wait.
They just wait.
An entire complex animal vitrified and paused. And the recovery mechanism is equally mind-bending to me.
Oh, it's incredible. If you take a ton that has been sitting in absolute zero for 10 years and expose it to a single drop of liquid water.
The sugar glass just rapidly dissolves.
Yep. The water rushes back into the cellular structures, the scaffolding rehydrates, the metabolism restarts, and literally within hours, the tardigrade uncurls its legs and just walks away.
Which is why this extreme terrestrial resilience naturally prompted astrobiologists to ask the ultimate question, right?
Of course. If they can survive the absolute worst Earth can throw at them, what happens if we remove them from Earth entirely?
Right. So scientists actually subjected tardigrades to low Earth orbit vacuum experiments.
They did. They took tons, placed them on the exterior of a satellite, and exposed them directly to the open vacuum of space.
Which means they are facing a simultaneous barrage of the worst extremes in the universe.
All at once.
They are dealing with the absolute vacuum, which rips moisture from cells and boils fluids. They are facing the extreme temperature fluctuations of space. And most destructively, they are exposed to unmitigated solar ultraviolet radiation and cosmic rays.
And that radiation is the true killer.
Because on Earth, our magnetic field and our thick atmosphere deflect the vast majority of this ionizing radiation, but in orbit, fully exposed. That radiation tears straight through organic matter, physically snapping the bonds of DNA.
It does. But astoundingly, when these tardigrades were retrieved, brought back into the lab and rehydrated, a significant portion of them fully revived.
That is just insane.
They repaired all the cellular damage, they resumed their normal metabolic activities, and successfully laid viable eggs.
They survived the vacuum of space, completely unprotected and reproduced. The key to surviving that orbital exposure has to lie in how they manage the radiation damage, right?
Exactly, because the radiation does shatter their DNA.
Right, the glass matrix protects the physical structure, but ionizing radiation still breaks the actual genomic sequence.
It snaps it right apart. Their survival hinges entirely on the rapid activation of a hyper-efficient suite of genetic repair enzymes the moment they rehydrate.
So the moment water re-enters the system.
These enzymes flood the nucleus, they identify the fragmented ends of the shattered DNA, and they splice the genome back together with flawless precision before the cell even attempts to divide.
Flawless precision, which is a perfect segue because that capability brings us to the absolute champion of genetic repair.
Ah, yes.
A bacterium known as Deinococcus radiodurans. If the tardigrade is the multicellular generalist, Deinococcus radiodurans is the unparalleled specialist in surviving extreme ionizing radiation.
It routinely survives acute radiation doses thousands of times higher than the lethal limit for human beings.
Thousands of times.
Yes. You see, gamma radiation acts like microscopic shrapnel. It blasts through the cell, severing the sugar phosphate backbone of the DNA double helix.
And in a human cell, just a few of those double-strand breaks can trigger apoptosis, right? Programmed cell death.
Right. The cell realizes its genetic blueprint is deeply compromised and just shuts down to prevent cancer.
But when Deinococcus radiodurans is exposed to massive gamma radiation, its genome doesn't just suffer a few breaks.
No, it is utterly obliterated.
The single circular chromosome is shattered into hundreds of disjointed, tiny fragments. It is a complete genetic explosion.
A total mess.
So how on earth do you recover from a scattered blueprint? How do the repair enzymes possibly know which fragment connects to which?
Because if you splice them back together in the wrong order,
you create massive mutations and the organism dies anyway.
Exactly. The secret lies in a highly unique structural adaptation. Deinococcus radiodurans does not allow its shattered DNA to just float freely in the cytoplasm.
Okay, what does it do?
It maintains its genome in an intensely compacted, highly organized ring structure, a tightly bound torus.
A torus, like a donut shape.
Yep. So even when the radiation violently severs the DNA in hundreds of places, this physical scaffolding holds the shattered pieces in their exact original alignment.
Oh, that makes sense. It's like keeping a fragile, complex face inside a tightly wrapped, heavy-duty net.
That's a great visual.
If you smash the vase with a hammer, the pieces are completely broken, but the net prevents them from scattering across the floor. Every shard remains physically suspended directly next to the shard it's supposed to attach to.
The physical proximity is perfectly maintained. So when the radiation event subsides, the bacterium's ligase enzymes, which are like the biological glue, can just rapidly move along the ring and stitch the fragments back together in the precise original sequence.
That is beautiful.
It can reconstruct its entire shattered identity within a matter of hours, totally error-free.
Now, this creates a really fascinating evolutionary puzzle.
It does.
Because evolution is driven by natural selection, right? An organism develops adaptations in response to selective pressures in its immediate environment. But Earth's surface has never naturally exhibited radiation levels anywhere near what Deinococcus radiodurans can withstand.
Right. There are no natural nuclear detonations.
So why would an earthly microbe invest massive metabolic energy into evolving a defense mechanism against a threat that doesn't even exist?
It's an elegant example of evolutionary cross-adaptation.
Okay, explain that.
The extreme radiation resistance did not evolve to combat radiation at all. It evolved as a direct byproduct of the organism's need to survive severe, prolonged desiccation.
Dehydration strikes again.
Yes. When a cell undergoes profound dehydration over really long periods in an arid desert environment, the lack of water induces massive oxidative stress.
And what does that do to the DNA?
That stress results in double-strand DNA breaks that are chemically identical to the damage caused by high-dose gamma radiation.
Oh, wow.
So Deinococcus radiodurans evolved the compacted ring structure and the rapid response repair enzymes simply to survive long, brutal droughts here on Earth.
So by evolving an ultimate defense against terrestrial drought, it just inadvertently pre-equipped itself with a biological shield capable of withstanding the radioactive vacuum of space.
Exactly. It's a terrestrial survival strategy that accidentally yields cosmic resilience.
And this bridges perfectly into the theory of panspermia.
It really does. Panspermia suggests that life on Earth may not have originated in some isolated primordial soup here on the surface.
Right.
It posits that microbial life, or at least the complex organic precursors to life, can be transported across interplanetary or even interstellar distances, trapped within meteorites, comets, or cosmic dust.
Like if an asteroid impacts a life-bearing planet with enough force, it ejects tons of rock and debris out into space.
Yeah, exactly.
And if organisms like tardigrades or Deinococcus radiodurans are nestled deep within the pores of those ejected rocks, well, we know they possess the biological hardware for the trip.
They absolutely do.
They can enter a ton state to survive the vacuum, they are immune to the freezing temperatures, and they have the genetic repair mechanisms to shrug off the cosmic radiation during the journey.
They could theoretically drift through the void for millennia, survive atmospheric entry on a totally new world, rehydrate, and seed a completely barren planet.
The low Earth orbit experiments didn't just test resilience. They functionally proved the biological viability of panspermia.
It radically shifts our perspective on the fragility of life.
It really does. I mean,
Yeah.
we've looked at extremophiles thriving in oceans, ice, and acid, and we've seen them survive the vacuum of space.
Right.
But if radiation and cosmic impacts are the ultimate sterilizing forces on a planetary surface, where is the ultimate safe room? Like, if you want to ensure the uninterrupted continuity of life on a volatile world, where do you go?
You go deep underground.
Which leads us to the endoliths, the architects of the deep biosphere. Endo, meaning inside, lith, meaning rock.
And we are not talking about a shallow cave system here.
No, we are talking about ecosystems residing miles beneath the continental crust and the abyssal ocean floor, occupying the microscopic fissures and pores within solid igneous rock.
These are environments completely sealed off from the surface world. There is no sunlight, no atmospheric gas exchange, and no descending organic matter.
It is an ecosystem entirely divorced from the surface biosphere.
Yes, and because they exist in such extreme isolation with incredibly limited resources, their biological clocks operate on an entirely different scale.
The pace of life down there is almost incomprehensible to us. I mean, some of these endolithic colonies divide